In class, we're covering 4 different types of inheritance: complete dominance, incomplete dominance, codominance, sex-linked traits, and we're also looking at pedigrees, which include all of these. We are going to look this time at incomplete dominance. Here is our problem. This is an incomplete dominance problem. Tail length for some cats shows incomplete dominance. Long tails, medium length tails, and short tails. Construct a Punnett square and give the genotypic and phenotypic ratios for a cross of, let's just underline, long tail and medium tail cats. Okay, if you haven't seen the steps video, go and find the video that shows you these steps I'm going to take, because I outline them slowly for you. I am going to go through them pretty quickly, but I am going to show you 4 steps that you should all do while you're learning how to do Punnett squares. Step 0 is the setup. And that means we're gonna create a key for ourselves. We're gonna list all the genotypes, and for this I am going to use T. And there's a couple ways you can label incomplete dominance. I'm going to do it one way and I will show you another way, okay? But for now I'm going to use T and S. TT is going to be long tail, and TS will be a medium tail, and SS will be a short tail. Okay, now the proper way to actually write that is to put them as a superscript. You'd go like this, and this means you're looking at incomplete dominance. Whenever you put it as a superscript over a C, that tells you you're looking at incomplete dominance. It's the kind of standard way of writing it, but it can be kind of confusing, right? So this is really how you're supposed to write it, but we're going to do it this way. Okay, this is homozygous for large T allele, okay? It's not necessarily dominant because we're looking at incomplete dominant, but it is homozygous. This is heterozygous, and this is homozygous for the short allele. I write the words so that I start to get to be really familiar with them, and I want to do it every time so that I will one day not have to even think about it. Step 1 is where we actually figure out the parents, which is the most important. So the parent cross. And we are going to look at long tail and medium tail. We know this one is long, so we'll actually make this a circle, we'll make this a dot, and we'll make this a star. And this is long and a medium, so we're going to cross these two. TT times TS. And remember, this is really the way you do it, but I'm getting rid of the superscript just to make it a little bit more easy to read. Okay, 2 is the Punnett square. Okay, and I like to put this on this side, T, T, DS, right? And then we're just going to do the Punnett square. So this goes here and here. This goes here and here. And then we bring this T down and S. Now let's find the phenotypes for each genotype. These are the genotypes, the types of genes. Let's look at the phenotypes. We have TT. We look at our key here, and we can see that we're going to circle these. Circle, circle, because these are both long. And TS is what we have here, and this is medium, and we're going to mark those with a dot. We have no short, which would be star, but we don't have that. 3 is to list the genotypic ratios and the phenotypic ratios. Genotypic ratio, phenotypic. Do you guys know what the difference is between those two? Genotypic is going to look at the genes, and pheno is going to look at the physical traits. Okay, so we just look at the possibilities here. So we're going to write the possibilities. These are the possible genotypes, genes. Okay, let's see here. TT, and okay, and let's do this first. So let's just count. We have 2, 2, and 0. Am I right? So we have 50% TT, 50% TS. See, when you have the ratio, you also find the percent. All right, phenotypically, we have three things that could happen, right? Long, medium, and short. Okay, let's count how many long, and we circled the long. So we have 2 circles. Pretty easy if you use the key symbols, you can't mess it up. If you have the medium, we put a dot, and so we see 2 dots. And do we have any stars? Any short? No. So once again, it's 50-50. % long, 50% medium. And so we have found the phenotypic and genotypic ratios for the long and medium. Let's recount. Let's do our 4th step, which is to check backwards. Did we count these correctly? I see 2 circled and 2 dots, 2 and 2. I see 2 TTs and 2 TSs. So we counted those correctly. Did we mark these right? We have the mediums here with the dots, TS, TS, and TT, TT, we have 2. So we did that correctly. There's 4 checks. And then did we move the T's in correctly? And did we basically do the Punnett square correctly? Check. And lastly, the parents. And so this is the most important. What do we have here? We have a long-tailed cat and a medium-tailed cat. Without even looking at the problem, try to say what you're crossing. Long-tailed cat, medium-tailed cat. What do they want? We go up and find it. Long-tailed, medium-tailed. We have it correct. That's how you do it. We have now done incomplete dominance using all the steps and showing our work. Good luck. And if you do not understand this, find somebody, ask some questions. Have a great day.